Welcome to the Bioprocess Pulse, a biomanufacturing and fermentation technology podcast focused on what truly drives industrial progress. This episode covers 13 to the 19th of February 2026, spotlighting real breakthroughs, scale-up realities, capital signals, and regulatory developments shaping the biotech landscape. Each week, we examine what moved the needle, what held steady, and what quietly creates competitive advantage. From upstream pathway engineering to downstream economics, from reactor design to compliance innovation, we turn technical change into practical insight. This is not about chasing trends. It is about identifying what works at scale. Let's dive into this week's Pulse.
Welcome back to the deep dive. Today we're focusing on a really specific slice of time that honestly, it looks like it's going to be pretty pivotal for the bioeconomy.
Yeah, it really does.
We're unpacking the Bioprocess Pulse Report. This covers the week of February 13th to the 19th, 2026. And, it was just a massive week.
It was, and not because of like a flashy IPO or some celebrity endorsement, right? It was massive because the actual machinery of the industry.
Biology and hardware?
Exactly. The biology and the hardware just seemed to click into a higher gear. It felt like a week of convergence.
Yeah, because often in this space, we get so much noise. We get these press releases about, saving the world with microbes or these wild targets for 2050.
Or those really cool concept art renderings of bioreactors in the desert.
Right. But this week, we actually saw genuine signals. We saw engineering data that maps directly to the balance sheet. And that's the distinction we need to make right up top for you listening. We are looking for what is actually deployable right now.
That is our whole mission today. We need to be the filter for you. We're separating the signal from the noise.
Because we're talking about a potential revolution in yeast engineering that sounds like it's finally ready for the big stainless steel tanks. Not just the glass flasks in a lab.
Right, and we've got AI tools slashing red tape and market projections hitting that $250 billion mark. But when you see a headline like 39% yield increase or zero-shot AI predictions, your immediate reaction has to be skepticism.
Does this actually work at 100,000 liters? Does the physics hold up when you add real heat and pressure?
Exactly.
So let's just jump right into the star of the show. And this wasn't a press release from a Silicon Valley startup. This was a fresh paper out of China that has the whole metabolic engineering community completely buzzing.
Oh, they are absolutely buzzing.
They've developed a new strategy for Saccharomyces cerevisiae, good old baker's yeast. And it seems to solve a problem that's been, honestly, a migraine for engineers for decades.
It has. And to understand why this matters, you really have to understand the tension inside a yeast cell. Yeast is the absolute workhorse of the industry. It's a GRAS host, generally recognized as safe.
Right. We've been using it to make bread and beer for millennia.
Exactly. We know how to grow it. But historically, if you wanted yeast to produce phenolics, specifically compounds like tyrosol, which are these really valuable antioxidants and precursors for other chemicals.
You basically ran into a wall.
A massive wall. It's a resource war inside the cell. The yeast basically decides it would rather survive than do your bidding. Producing these phenolics creates a huge redox imbalance.
It just uses up all the precursors.
Right, all the raw materials the yeast actually needs to build its own cell walls and proteins. And on top of that, the product itself, tyrosol, is usually toxic to the yeast at high concentrations.
So you get what they call toxicity crashes.
Yeah. You start the fermentation, the yeast grows, it starts making the product, and then boom, the culture just collapses. And dead cells don't make money.
Which is the nightmare scenario for a founder. You spent the money on the sugar, the electricity, the labor, and you just end up with a tank of expensive soup.
Yeah.
So what actually changed this week? Let's talk about this new strain, ST82.
Strain ST82 is the breakthrough. So in a 5-liter fed batch setup, and note that is a decent pilot size, not a tiny test tube, they achieve tyrosol titers of approximately 3.2 grams per liter.
Okay, let's just pause on that number for a second. 3.2 grams per liter. Now, if I'm looking at something like ethanol, that sounds tiny. Ethanol hits way higher concentrations. Why is 3.2 so impressive here?
Well, context is everything. Ethanol is a primary metabolite. Yeast naturally wants to make it.
Yeah.
Tyrosol is a complex secondary product that actively fights the yeast's natural instincts. So in this specific context, 3.2 grams per liter is huge. It's a 39% jump over previous benchmarks.
Yeah.
But the number itself isn't even the most exciting part. It's how they actually got there. They didn't just tweak one gene. They did this multi-round flux overhaul.
Flux overhaul? That sounds intense. Break that down for us.
Think of the yeast's metabolism like a really complex city traffic grid. Usually, engineers try to open up one new road to get carbon to a specific destination.
But if the highway isn't wide enough, or if there are too many off-ramps leading somewhere else,
Yeah.
you just get a bottleneck.
Exactly. You get traffic. So this team rewired the entire shikimate pathway. They amped up the tyrosine pathways, that's the main highway. They cut out the competing routes, the off-ramps that were stealing resources, and they fine-tuned the oxidation processes so the traffic didn't overheat the whole system.
And the absolute kicker here, from what I was reading, is that they did all of this without using plasmids. That seems to be a major sticking point for the industrial crowd.
It is the crucial detail if you're looking to scale. Plasmids are basically like sticky notes you put in a textbook. They have the information, but they can just fall out.
Right, they're just extra loops of DNA.
Yeah, they're great for quick experiments in the lab, but they are incredibly unstable. To keep them in the cell, you usually have to use antibiotics, which is expensive and messy at scale. But this new strain has these changes genome integrated.
So it's hard-coded. It's printed directly in the book, not just on a sticky note.
Precisely. The instructions are hard-coded into the yeast's main DNA. No plasmids, no expensive antibiotics, no fancy chemical inducers needed to switch them on. The strain is genetically stable from day one.
Which brings us directly to the industrial implications. Because, you can have the best strain in the world, but if it requires some gold-plated custom bioreactor to grow, it's completely useless.
And that's why this is such a clear signal. This setup is perfectly compatible with standard aerobic stirred tank infrastructure. You don't need to build bespoke hardware. It fits within standard oxygen transfer limits, what we call cake.
Let's actually dig into that term, cake lobby, because it gets thrown around a lot in these reports. How should you visualize that if you're listening?
Think of Kaliala like the lung capacity of your tank. If you have a high-density culture, it's like a crowded room sucking up oxygen. If the lungs, meaning the impeller stirring the liquid and the sparger blowing in the air if they can't keep up.
The cells basically suffocate.
Exactly. But this strain doesn't run a marathon that leaves it gasping. It works comfortably within the tank's breathing limits.
And I noticed the report mentioned rheology as well. That's viscosity, right?
Yeah.
The actual thickness of the liquid.
Right. Sometimes when you engineer these microbes to make complex molecules, they get stressed. They start producing slime or these weird byproducts that turn the whole broth into a thick goo.
A rheology nightmare.
A total nightmare, because you can't stir it and you can't get oxygen to the cells. It effectively destroys the batch. But strain ST82 maintains moderate viscosity. It flows like water, not ketchup.
So for the founders listening right now, this creates a real moat.
Yeah.
If you have this strain, you're way ahead of the game.
You really are. It's a GRS chassis, so your regulatory approval is easier. It uses standard hardware, so you don't need exotic CapEx. You can take this platform, which is hardened at the shikimate node, and pivot to produce all sorts of derivatives.
And for the engineers, the problem sets completely change. You're not fighting to keep the DNA in the cell anymore.
Exactly. The focus shifts. Now you just focus on density ramps. How many cells can I jam into this tank? And in situ removal. Because 3.2 grams per liter is the magic threshold where downstream processing actually becomes economically viable.
Right. Let's talk about the downstream side, the DSP, the boring part after the fermentation where you actually have to extract the product. Why is 3.2 the magic number?
It's purely economics. If the concentration is too low, you're pumping thousands of liters of liquid through really expensive filters and distillation columns just to get a tiny handful of product out.
You spend more energy extracting it than it's worth.
You bleed money. But at 3.2 grams, you can use standard adsorption or solvent trains profitably. It's the literal tipping point between a fun science project and a viable business.
Okay, so we've got the wetware sorted, the yeast is looking good, but there was another major accelerator in the report this week, and it involves everyone's favorite buzzword, AI.
Of course.
But this isn't just asking a chatbot to write a marketing email. This is AI deep in the lab and, surprisingly, in the compliance office.
Yes, two very different but equally massive applications surfaced. Let's look at the lab first. MIT released news about a large language model specifically designed to predict codon and promoter optimizations for yeast.
So instead of predicting the next word in a sentence, it's predicting the best genetic sequence to get the yeast to express a specific protein.
Exactly. DNA has a grammar, just like English. This model understands that grammar, and it allows for zero-shot predictions.
Zero shot, meaning you get it right on the very first try. Is that actually realistic?
In this context, it's revolutionary. Usually, strain engineering is a brute force screening cycle. You build 1,000 variations, test them all, find the best one, learn from it, build another 1,000. It takes months.
Right.
This model acts like a sniper. It drastically reduces those experimental cycles. We're moving from months of trial and error down to just weeks of verification.
That is massive for a startup's burn rate. Time is literally money there.
It is. Faster development cycles, stronger IP positioning, and it enables high-density fermentation without those viscosity spikes we talked about because you optimize the code so the cell runs efficiently from day one.
But I have to admit, the AI news that surprised me the most was on the regulatory side. eBiotech launched a platform for GMP compliance. And usually AI and FDA regulations do not mix well.
They really don't.
Regulators like paper trails, they like human signatures.
They love them, and GMP, good manufacturing practice, is the absolute bane of many creative scientists' existence. But it is non-negotiable if you want to sell a product for human use. Historically, it's stacks of binders and manual validation.
So how does an AI help with that? And do regulators actually trust it?
That's the billion-dollar question. What eBiotech is doing is embedding adaptive reasoning into the workflow. So it's not just a static checklist. It's an AI that understands the context of the regulations across multiple jurisdictions.
So if you change a parameter in the lab, the AI instantly flags which exact reports need to be updated for the FDA in the US and maybe the EMA in Europe.
Correct. It streamlines all that multi-jurisdiction documentation. And here's where it gets really interesting for the market. This is a weapon for agile mid-sized companies.
Because the legacy pharma players are stuck on their rigid systems.
Exactly. If a mid-sized company can integrate this digital quality assurance early, they can move so much faster, it automates the boring but hard stuff.
So we have the biology dialing in, we had the software speeding things up. Let's zoom out to the macro view, the hardware market, because the report drops some eye-watering numbers here. The global bioprocessing market is projected to grow from about $93 billion in 2025 to $277 billion by 2035.
That's an 11.5% compound annual growth rate. It's aggressive, but honestly, it aligns with what we're seeing on the ground.
That is a lot of steel. What's driving all that growth? Is it just people building more of the exact same tanks?
It's more tanks, but it's also better tanks. The growth is driven by bioreactors, but also advanced filtration systems and modular hardware. And there's a huge specific focus on inline sensing.
Inline sensing. Why is that the hot topic right now?
Because we are moving toward long duration runs.
Yeah.
If you're running a fermentation campaign for days or weeks, things drift. The pH drifts.
Yeah.
Metabolites foul the sensors. Variability just creeps in.
Right.
Think of it like a self-driving car. If you have to stop the car every 10 miles to manually check the map and calibrate the speedometer, you're never getting anywhere. Inline sensing allows the tank to essentially drive itself, correcting for pH and nutrient levels in real time without stopping.
Which completely reduces the human error factor.
And the contamination risk. Every single time you open a port to take a manual sample, you risk letting a stray bacteria in. Inline sensing keeps the system closed.
We also saw some capital movements that back this market confidence up. Novo Nordisk is expanding their facility in Ireland for Wegovy pills. Now, I know that's big pharma, not a scrappy startup, but does that signal trickle down to the rest of the industry?
It definitely signals confidence in manufacturing scale. Technically, it isn't a fundamental process shift. They aren't reinventing the wheel. But the fact that they are pouring billions into manufacturing scale for obesity biologics says, we believe we can make this at massive scale reliably.
It stabilizes the supply chain for everyone else who needs those parts.
Exactly. And the report noted something else really interesting on the scale-up watch. There were no new lab-to-commercial transition autopsies this week.
No big public failures.
Right. Silence is golden here. It means tech transfers held steady. We didn't see any high-profile crashes where a company promised the moon and just delivered a puddle of sludge.
Nobody had to explain to a board of directors why their 50,000-liter tank smells like rotten eggs.
Exactly. No confirmed economic boundary collapses this week.
We will take the quiet wins. Now, we have to pivot to the geopolitical angle because this is something we cannot ignore. China released its 15th five-year plan, and the numbers are staggering.
This is a massive policy signal. The stated goal is to accelerate biomanufacturing toward approximately 1.8 trillion yuan by 2030.
That is a lot of zeros. But beyond just the money, what's the actual strategy? Because China doesn't just throw cash around randomly. They pick a lane.
They have picked a lane, and it's a very smart one. The key phrase to pay attention to is waste to value. They are putting incredibly strong policy backing behind microbial systems that can essentially eat waste.
So taking trash like agricultural runoff or industrial byproducts and turning it into valuable chemicals.
Exactly. And this fundamentally changes the economics of the whole industry. In
Yeah.
the West, we are often obsessed with clean sugar, dextrose, sucrose. It's easy to work with, but it has a hard cost floor.
Right, sugar isn't free.
No. But if China succeeds in scaling systems that run on impurity-rich feedstocks, meaning the food you give the yeast is dirty, variable, and cheap, they create a massive competitive advantage.
It effectively lowers the cost of goods sold to near zero for the input.
Right, but it's incredibly hard engineering. You need robust strains like that ST82 we discussed earlier that don't just crash when they encounter a random toxin in the trash. It connects right back to the biology. If you can design a strain that eats garbage and makes gold, and you have the government paying for the steel to put it in...
That's a winning combo. But it sounds like a strategic opening for agile Western companies, too.
Yeah.
If you can handle dirty feedstocks and use AI to compress your compliance, you can play in that global market.
Precisely. It's a call to action for the engineers out there. Stop cuddling your yeast with pharmaceutical-grade sugar. Teach them to eat street food.
Teach them to eat street food. I love that. Now, before we wrap up, we have to do our Hype Buster segment. This is where we bring everyone back down to earth. And the report this week took a pretty hard look at precision fermentation.
Yes. This is a topic that generates a ton of excitement, and rightly so. We all want animal-free dairy and egg whites without the chickens. But there's a viral claim out there that precision fermentation is just a universal molecule printer.
Right, the whole Star Trek replicator idea. You just program a microbe to make anything you want, milk, meat, leather, jet fuel, and it'll replace the traditional agricultural way of doing it overnight.
And that is exactly where we need the cold shower of physics and economics. The report heavily emphasizes that the industrial constraints are very real. It's not magic, it's thermodynamic.
You have the substrate costs we just talked about.
Right, and you have utility bills, the electricity to spin the massive impeller, and the water to cool the jacket of the tank.
Because fermentation creates heat.
Massive amounts of heat. A fast-growing yeast culture is basically a biological fire. You have to constantly cool it down. If you can't remove the heat fast enough, the physical limits of the tank cap your production speed. And then you have the downstream issues. Resin fouling.
Resin fouling. Explain that one for the audience.
So, to purify your target protein out of the broth, you often run the liquid through beads resins that chemically grab the product. But if your broth is full of other junk like dead proteins, cell walls, DNA, those beads just get gummed up.
They stop working.
Right, and cleaning them or replacing them is wildly expensive. If you're trying to make a bulk commodity protein, something really cheap like casein or weigh the scale physics, and the cost of replacing those resins will absolutely destroy your margins.
So what's the verdict? Is precision fermentation a dead end?
Not at all. It just needs economic realism, not press release optimism. Precision fermentation is best suited for high-value niches. Medicines, specialized enzymes, complex flavor molecules, lactoferrin.
But not commodity displacement.
Exactly. It is not going to displace cheap commodities like soy protein or palm oil anytime soon. So for the founders listening, you have to anchor your projections in realistic, tighter assumptions and actual willingness to pay. Don't pitch a commodity product at a pharmaceutical price point.
And for the engineers, you need to model full chain economics and fouling at the 50 to 200 cubic meter scale. Don't just show us what happens in a 2-liter flask. Show us the math for a giant steel tank running for 3 weeks.
Exactly. Large-scale physics and economics remain the ultimate gatekeepers here.
It sounds like the theme of this whole deep dive is integration. You can't just have the strain, or just the AI, or just the tank. You need the strain to fit the tank's KLA, and the AI to make sure the strain is compliant with the FDA.
That's it. We're moving from the Wild West phase of synthetic biology straight into the Industrial Revolution phase. The tools are standardizing, compliance is getting automated, the strains are getting robust enough to handle the real world.
So pulling this all together for our listeners, what does this week's Pulse actually tell us?
The bottom line is early-stage flux rewrites and AI-driven tools are showing actionable promise. We have genuine upstream progress with the yeast, we have AI accelerating both strain design and regulatory workflows, and we have a hardware market that is booming but governed by strict physics.
And we have the geopolitical layer, like China's waste-to-value push, that could quietly reshape speed to market.
Right. It's a convergence of better biology, smarter software, and realistic hardware growth. But we have to keep a very close eye on this scale-up watch.
And that brings us to the final thought to leave you with today.
Yeah. I want you to think about this. We saw no major tech transfer failures this week, no big scale-up disasters reported, but are we just in the calm before the storm?
What happens when these ultra-optimized strains hit the real world?
Exactly. As these new genome-integrated yeast strains like ST82 actually hit the 100,000-liter tanks, will the physics hold? Will the multi-round flux overhaul survive the sheer physical stress of a giant industrial impeller over a 3-week campaign?
That is the multi-million-dollar question. And I guess we'll find out soon enough. But for now, the signal is strong.
It's very strong.
And that is a perfect place to leave it. Look for the signal in your own data this week. Thanks for diving in with us.
Catch you next time.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.